the Science Cafe with your host, Chris Smith.
Hello, hello, hello. Hi, everybody. Welcome to the museum. Welcome to the Science Cafe for tonight's program. I'm glad you all decided to come out. Did anybody have trouble getting to the museum? Yeah, crazy stuff, right? So I can inform you on what's going on a little bit because I went and checked on it. So earlier this afternoon, apparently a water main broke somewhere up there, and all that extra water came rushing underground this way and then came up out of the storm drains at the Salisbury-Jones Street intersection over here. And doing that washed everything out. The road buckled, and so Raleigh police came out and shut the road down, and now it looks like they're probably going to be working on it for a while. So we'll see if I can get to work tomorrow. Do I have to come if I can't get around the... Oh, I have to... Okay. It's a science museum. Why wouldn't I want to come to work, right? We get to do all kinds of fun and cool and interesting stuff here at the museum all the time. There's always great programs going on. Even tomorrow at 11 o'clock here in the museum, we're going to have a special presentation with a guest who's going to be talking about the importance of museum collections at 11 a.m. So if that interests you, come on out. It'll be in the Daily Planet Theater, talking about what makes museum collections fun and special. And then tomorrow at 1:30, I get up on stage and I do my own little show and dance, my own little trivia show. So if you're good with your science trivia, I do my own little fun thing in the Daily Planet. That's every Friday, 11 o'clock and 1:30. You can come check out a cool program. At least I hope you think it's cool, because I do it. But right here in the museum. And then stuff like that happens every day. In addition, there are fun special events going on all the time, right? Science cafes happen here in the Daily Planet Cafe every Thursday at 7 o'clock. So you can come out every week and learn something new about science, technology, engineering, mathematics, and meet the interesting and exciting people who are doing that work. Coming up on February 23rd, everybody's got a little bookmark on their table. Several of you may have heard me talk about this already, but we'll have authors John Grisham and John Hart on stage at Maymandy Concert Hall talking about creativity, imagination, and writing their novels. And you'll also hear just a little bit from museum paleontologist Lindsay Zano. She's going to be talking about creativity and telling stories when it comes to reconstructing the past, the work of paleontology and the work of many other sciences. So there's a lot of cool synergy happening between science and the humanities and the arts going on with that February 23rd event. So get tickets for that one. I'll be there. I got my tickets today, so check it out. Maybe you can sit with me and everybody will think you're extra special. No, that's not true. For tonight's program, we're going to be talking about fighting pollution with designer microbes, which harkens back to me. Did anybody go see the Secret World Inside You exhibit that was here in, I think, 2016? Yeah, a few, or 2017. Yeah, a few folks. Awesome. So microbes everywhere. We live in an invisible world that quite nearly dictates everything we do. Like, we're alive in the Science Cafe, enjoying the food and the drinks here because we have microbes on us and inside us that help us do that. But there are microbes everywhere, all the time, everywhere. I even think there are microbes in space. At least I've heard of ones that can survive in space. So microbes are everywhere, an invisible world. How can they help us, at least the ones that aren't living on us and inside us that help us survive? What about the ones that are out there in the environment? How can we recruit those organisms to help us with our daily lives? And it sounds like in this case, clean up after us, because we know that humans are a messy bunch living on planet Earth. Our guest tonight for the Science Cafe is Dr. Claudia Gunsch. She's civil and environmental engineer at Duke University and associate professor there. She won an award from the National Science Foundation for her work in 2009, I read, and then 2 awards, including one from the American Society of Civil Engineers just in 2016. So I think we got a good one tonight. Put your hands together and welcome to the stage, Dr. Claudia Gunsch.
All right. Well, Good evening, everybody. Thanks for having me. And today, I thought it was very fitting that there's a water main break because, of course, civil engineers would solve that problem. And there's water and there's environmental engineers. So maybe it was special for me. So I'm Dr. Claudia Gunsch from Duke University's Department of Civil and Environmental Engineering. I wear a lot of hats, and my career has really taken me in a lot of different ways. All of my training is in civil engineering, but as I went through my graduate career, I was exposed to microbiology and genomics, and this was right when genetics was really exploding, and there was a lot of interesting research. And of course, I never like to go in one direction, which is why I'm in research. And so what I'm going to talk to you today about is really how I've integrated all of that into my research. You really do have to get really close to this. All right. So as was alluded to, we have microbes everywhere. And so you've probably heard of the term microbiome. Microbiomes are really microbial assemblies. And there's been a lot of work that has been on the topic of microbiomes. And as was alluded to, any surface, any place where you go, there will be microbes. So I always tell my students and people that are listening to me that microbes really rule the world. And so this is showing a cell phone and thinking about your cell phone and how you interact with your cell phone with your hands or up to your face, and thinking about how you might transfer microbes from one surface to another. So your cell phone will have a certain microbiome, so a certain microbial assembly, and somebody else's cell phone will have a different one. Similarly, if you go out into the ocean, there's more microbes that are present there. They're different microbes, but all of those microbes have a certain purpose and are there to carry out a specific ecological function. Now, thinking about another place where you might run into microbes. So here I'm showing a roadside where there's lots of cars, and you can imagine that the things that are present in that atmosphere are going to be very different than your cell phone environment or that ocean environment. And so each of these are going to have signature microbial assemblies that are present and are carrying out different roles. So we're familiar with the human microbiome. It's amazing to think about how each part of our body has different microbial assemblies associated with them. And we're now capable really of going, and based off of the microbial signature that we find, we're able to identify which organisms are there and start thinking about how we might be able to engineer those microbial assemblies. So, is this still on? Is this still on? Okay. So, in the human microbiome, when we think about the human microbiome, there are more microbes in the human body, a number estimated to be in the trillions, than there are stars in the Milky Way. So, to put this into perspective. Same if you think about just the vast quantity of organisms that are present, all of the microbes combined weigh about as much as your brain. And so again, if you needed any convincing that microbes are actually important and are carrying out important functions, just thinking about the sheer quantity of microbes. And similarly, the microbes are going to change when you're thinking about as somebody gets nervous, as they start sweating, there's going to be different signatures that are going to be associated with it. So this is with the human body. And so in the human body, we've now come to the point where we know what microbes are present, and we can get a pretty good understanding of what's happening. And really, and these advances have enabled us to develop certain precision medicine applications. Now, where I'm interested in is understanding when there are interactions between the environment and humans, how those are altering these different microbiomes. So you could think about the air microbiome, the food microbiome, and the water microbiome. So microbes out in the environment are responsible responsible for shaping all of these ecosystems. So they carry out the carbon cycling, they carry out nitrogen cycling, and those are very important functions that when we alter that environment will alter their ability to carry out those functions. So microbiomes have global impact. There's estimated to be 1 trillion species on Earth, and the vast majority of these we have no idea. We haven't been able to culture them, and so this is where a lot of my work lies, understanding how humans impact nutrient cycling, how climate change also is going to alter these microbial functions. So thinking about how we affect these ecosystems. So I'm showing a picture up here of the spill, the mine waste spill, and you might have heard about this one that happened several years ago, where somebody had gone out and actually was sampling and released a bunch of waste in the environment. And so you can see in the top picture, maybe you can see it. It's before the contamination actually happened. So it looks like a normal river, and then on the bottom the water has turned yellow. And so you can imagine when those types of events happen that there's a drastic signature that occurs. There is impact on the microbes that are present in those environments. Similarly, in oceans, here I'm showing, these aren't actually a pre and post picture, but just showing an ocean and all of the microbes that are present in there, and then when an oil spill occurs. And so my research and the work that we do is really at trying to understand what microbes are resistant and actually like to live in the environments after those contamination events have occurred, and trying to really take advantage of their nature, their natural capacity to be able to degrade or survive, at least in those environments. So there are many hazardous waste sites, unfortunately, in the United States, and if you went worldwide, there's even more. And so in the United States, there's approximately 5,000 waste sites, and here we're showing a couple of pictures of what those waste sites look like. You can imagine, based off of the pictures and what you're seeing here, that the contaminant profile that is present at each of these sites is going to be drastically different, and there's also going to be very, very different microbiomes associated with each of these situations. So just to put it into perspective, and you can't really see it right here, there's a map of this region, and actually we usually do it, of course I'm from Durham, from Duke, so usually we look at Durham, but most Americans, so a large number of Americans, so 53 million Americans actually live within 3 miles of major hazardous waste sites. And here we're talking about Superfund sites. So Superfund sites are sites that are actually have been turned over to the government, usually because a company may have gone out of business, and we are trying to remediate those sites. So this is a very big problem. There are sites that you might not even realize are contaminated sites, because there might be grass, or there might be, it just doesn't look aesthetically like it should be a hazardous waste site. So what the research that we do is termed bioremediation. And so bioremediation is really taking advantage of these microbiomes to degrade contaminants. And so the idea is very simple. We look for microbes that are adapted to grow under these difficult conditions. And in many cases, these organisms are capable of consuming the contaminants. So they're using the contaminants as their food source. And hopefully, if everything goes well, they can actually degrade the contaminant, turn it into new cell material, so new cells, and transform it into non-toxic end products. And so if in the best case scenario, we are turning it into cells and carbon dioxide that can then just be released into the air. So in many cases, there are microbes that are present at these sites, but maybe they're not working as effectively as they should be. And so this is what, so there's a couple different strategies that can be used. The first one is what is known as biostimulation. And so in the case of biostimulation, we look for microbes that are present. If we have an idea of the microbial function, so let's say we know that this particular gene, for instance, carries out a particular function, so it can degrade, let's say, TCE or toluene or gas or whatever your contaminant of interest is. If you can go and look for that particular organism, but maybe it's not degrading very well, you can develop a strategy to actually go and stimulate it by adding maybe some nutrients or something along those lines. If you do not have a microbe at your site that is capable of degrading your contaminant of interest, you can utilize a strategy known as bioaugmentation. Bioaugmentation consists of taking microbes out of, let's say, your lab, or you can also purchase them from companies that have a list of organisms that can degrade particular contaminants, and then inject it into your subsurface and trigger degradation in that way. So bioaugmentation often doesn't work very well. So you can imagine that if you're taking a microbe from a very specialized condition, let's say from your lab, you know exactly how to grow it in, let's say, a liquid, and you're putting it into this foreign environment that's very heterogeneous. So you can imagine soil is going to be, there's lots of different pockets of maybe oxygen or pockets of nutrients there. And so we want to understand how can we optimize those treatments to maximize the amount of degradation that's possible. And so we can combine biostimulation and bioaugmentation, but in many cases, those organisms don't actually survive in the environment. And so that, of course, as an engineer is very frustrating. And so what we've done to help with this is we can start now looking for these genetic signatures. And so I got my PhD back in 2004, and in 2004, the extent of what we could do really using genetics and looking for specific microbial fingerprints was shown up here on this gel. So what this picture shows, each one of those little black bands that you see actually represents a single microorganism. And so you could look for presence or absence of a specific microorganism, but you really didn't get much information. And so the way I describe it is, in the past, we were really data limited. And so we knew a little bit of information, but it was really difficult to do any meaningful design for very complex scenario. At present, where we become limited is data analytics. And so I know last week you had a talk about big data, and this is the challenge that we are currently facing. So we can get a fingerprint, a microbial fingerprint at a site. We can actually screen the entire genomes for thousands of different microorganisms at the same time. But remember what I said earlier, that there are 99.99% of these organisms, we don't even know what they do. And so while in the human body, we might have a good idea of what those microorganisms do and we can characterize them, when we go out into the environment and we have these very heterogeneous conditions, trying to make sense of that becomes very, very challenging. And so we are developing methods that are biostatistical methods and bioinformatic tools to be able to solve this challenge. And so I am an investigator on one of the Superfund centers that is funded by the National Institute of Environmental Health Services. And we have been working at a number of Superfund sites here in North Carolina. One of them I'm showing up here, the Holcomb Creosote site, which is a former site where there was a wood treatment that took place that has been heavily contaminated with a class of compounds known as PAHs, polycyclic aromatic hydrocarbons. And at this site, we have started identifying what fingerprints are present and trying to make associations, so trying to figure out which microbes associate with a certain class of chemicals. Oops. So, and so we're moving in the direction of precision bioremediation. And so the idea is that very much the way that in precision medicine, we are working towards probiotic applications, where for a specific disease or a specific ailment, you could take some type of cocktail of microbes to solve that problem, or at least begin solving that problem. Here, the idea is that if you have a specific contaminant present, and you know that you have certain environmental conditions, we can start developing models to really identify which microbes or what stimulation strategy we can use for that targeted site. And so this is just an example. It's a really busy figure right here. What I want you to get away from this is that there's, thousands of different microbes that are present. There's hundreds of different contaminants that are present at this site. And we can now utilize these models to really pinpoint which microorganisms we can go after. And so this was at this Holcomb creosote site. We're now able to really get all the way down very precisely to which microorganisms actually are enriched or enjoy being in those conditions, and devise these precision bioremediation strategies for biostimulation, bioaugmentation. And then I haven't alluded to this, but there's also mechanisms which we can talk about where we can actually transfer native genes from one organism to another in an attempt to devise better treatment technologies. So moving forward, really where we are heading is, now that we have this, we've developed this we're developing a better understanding of microbiome assemblies and developing models that are predictive in nature, we can start moving towards these precision bioremediation strategies to degrade specific contaminants under specific conditions. And so a lot of my funding I have to acknowledge from the NSF and NIH. And then I also want to acknowledge that we have a graduate training program called IBEAM that I direct. IBEAM stands for Integrative Bioinformatics, for investigating and engineering microbiomes. And part of that program is really at training the next generation of students to be able to undertake these types of analyses, because traditional civil and environmental engineers certainly do not get this type of training. So with that, hopefully I gave you enough food for thought, and that will generate lots of questions. Thank you.
Let's give her a round of applause. Thank you very much. All right. So if you've got questions, raise your hand. The way it works is that Katie and I will come around with microphones, and we'll get to as many questions as we can. We've got plenty of time to have a great discussion about bioremediation and the techniques that we talked about. So let's get started.
Great. Thank you very much. Great presentation. I wanted to ask you about something really speculative. You talked at the very end there about genetic augmentation of microbes. How far away are we from building a microbe from the ground up for these purposes? Is that like years away, decades away, or pure science fiction?
So, well, there's a lot in that question. So I'll say there's been a lot of research that has gone into kind of the minimalist bacterium. There's a name for it, but I'm not, I can't remember what it is off the top of my head. So I think we know what are the basic functions that are needed in a microbe, and really trying at minimizing the energy that is required and maximizing whatever application. So for energy production purposes, for instance, there's been organisms present, or organisms that you can use for that. My own perspective on that, and this is me speaking personally, we don't really understand the full extent of what may happen if we were to introduce one of those organisms into the environment. We know based off, we're starting to move towards these, predictive models where maybe if we know we augment or add a particular microorganism, that might cause this other microorganism microorganism to decrease, which might have some ultimate effect on function. So I think it could be potentially dangerous, or at least it's something that I'm not willing to take a chance on yet. Whether or not we'll ever get there, I don't know. The strategies that we are actually implementing is, there are genetic elements that are present in bacteria that are known as plasmids. And so plasmids are these extra chromosomal fragments of DNA. And so those are fragments of DNA that cells don't necessarily need, and they usually carry the genes for all these extra functions that microbes may need. And so it makes sense for a lot of the functions that organisms need to degrade contaminants, that those functions are actually present on these extra chromosomes, because usually there aren't contaminants present, and so they take up those functions under those specific conditions. So the strategies that we're developing along genetic bioaugmentation is where you can actually introduce an organism that maybe doesn't compete very well in the subsurface, but it might transfer just the genes that it actually needs until those contaminants are gone. And then once those contaminants are gone, those genes also disappear from that microbial assembly. So in that, we are much closer towards really being able to actually put that into place. Thanks.
So since you said you don't know a lot about the total range of microbes that are out there, is it easier to approach this from the point of view of what the sites themselves are like? So do the 4,700 Superfund sites in the United States, for example, fall into certain categories that would encourage you to create, I don't know, model microbial assemblies that work in those conditions, or are they all entirely different from one another?
Yeah, so that's a great question. The real answer to that is that we really don't know. So we don't have the microbial fingerprints for all of these, thousands of different sites. So I would say probably in a decade we will, and hopefully before, we'll be able to answer those questions as the cost of sequencing comes down and we can really get a better appreciation for that. So I think we kind of have to approach it from both directions. But as we build our databases and we know more about what are the specific interactions between these contaminants and the microbes, we'll hopefully get to exactly what you suggest. Yeah.
Hello? Yeah, okay. What do you do if you get a witch's brew of, say, polyaromatic hydrocarbons and organophosphates and maybe some arsenicals? And, I can't imagine a single organism going to thrive in that kind of mixed environment.
Yeah. So that's the $1,000,000 question right there. So in the introduction, right, before I was, while I was being introduced, right, it was pointed out that there are microbes everywhere. There are very few environments that exist where there are no microbes. And so what we're trying to do is identify which microbes, maybe they don't thrive under those environmental conditions, but they're able to survive under those environmental conditions, and then take advantage of their abilities. So here I talked about, polycyclic aromatic hydrocarbons, but there's also very high metal concentrations that are present, which are also going to be toxic to some of these organisms. So we're going at it from a couple different directions, and also I talked about bacteria specifically, but we're also going at this from utilizing fungi that can actually break down some of these compounds in association with the bacteria. So you're right, it's very complex because there are sites where there are, thousands of different things that are present. They're partially degraded, and then they are toxic, and not a single organism can degrade all of them. We really need to be able to stimulate a range, a cocktail essentially of microbes. But the example that I always give my students is Dihalococoides is no, wrong organism. Deinococcus radiodurans is an organism that was isolated, I think about a decade ago at this point, from a Department of Energy site that had very high levels of radiation. And so this organism is able to withstand, I'm wanting to say it's like 10,000 or 100,000 times higher radiation levels than that you and I would be able to survive at. And so it's really by understanding what is it about that organism that gave it that advantage, and being able to really utilize that to our advantage to engineer these microbiomes. And so under most conditions, there are going to be microbes, but really just trying to figure out how can we really stimulate those to the best potential.
So what kind of timescales are involved with using these techniques to treat a site? How much do you have to apply, and then how long do you have to wait?
Yeah, so that's a great question. That's another $1 million question. So it's going to take a long time. So this is one of the drawbacks to utilizing a bioremediation strategy as opposed to using a physical chemical treatment application. It takes a really long time. So typically, if you want to use this application, you need to, this strategy, you need to make sure there's no potential people downstream, for instance, that have groundwater where that contaminant might come into play. So looking at really what are the potential human health or potential routes that way. That being said, it is much more sustainable from a practical standpoint, where you're not actually digging up dirt and doing some type of treatment. So it's going to take a long time, but in the long run, it's actually a much more beneficial. So I think, you know, there's people on both sides of both sides of that. It's going to be very different, also depending on the contaminant profile. So some of these sites that we're working at, it's been decades and nothing has degraded. So it can take a really, really long time to do it. So it's all over the board.
My question was about the economics of your work, and where do the microbes come from? Are there microbe farmers?
The economics, so that actually bioremediation as compared to the physicochemical treatments is actually economically much better proposition, because you're relying on the microbes that hopefully are already there, and so you would just have to add the nutrients or oxygen or maybe some type of amendment. And so typically the amendments that we use are things that are pretty cheap, so like molasses. There's also some strategies that rely on activated carbon, so things that are pretty easy to do. In terms of the microbes, so there are microbes usually at a site that are just, they have evolved over time and developed either the ability to utilize that food source because it's the only food source that's around, or they've been isolated from another site over time. And so that's something you could naturally evolve an organism, and the lab would be another way of doing it. So there are companies out there that sell specific cocktails to remediation outfits. I don't usually work with those types of organisms. I'm usually more interested in which indigenous organism that are actually out there. But it's amazing. The first place as an environmental engineer where I go if I want to find a microbe is actually the wastewater treatment plant, because a wastewater treatment plant has a cocktail of things in it because we're constantly releasing a variety of different chemicals to the wastewater treatment plant. So that's usually the first place to go. But even just outside, just grabbing a soil, there's often the right organisms to do what we need. So.
Hi, yeah, I had a question sort of about the operations side of the treatment. I was curious about the amount of microbes that are required to treat a site. Like, is it just a few ounces of microbes, or is it like hundreds of gallons?
So It would be more in the hundreds of gallons. So typically, if you were to do this, you would probably grow them in some type of big reactor and then inject them into a subsurface. The other way that this could be done is to add a small amount of microbes and then add maybe a rich carbon, so like molasses or some type of sugar that would then allow them to grow under those conditions. So, but you do need a fair amount. The other aspect of this is thinking about where your contaminant is. So if it's a hydrophobic contaminant, meaning that it doesn't like to be in the water and it's attached to soil, then that contaminant may be in a pretty small location. But if it's in water and it's constantly migrating downstream, for instance, you would need more microbes, or you would need to think about a different strategy. So again, it's going to be very specific to your site, but you do need the more microbes you're able to get, the better off. At least it'll go faster, I'll say.
So I have a question. Is there any work being done to use the microbes to treat waste before it becomes part of a Superfund site? I mean, is there any work to try to use that before it becomes something that we have to fear in the environment?
Yeah, so there's actually a fair amount of work. I mean, so keeping in mind that once a site becomes a Superfund site, it's because there's been some type of release into the environment. So once it's released into the environment, then you could just do the bioremediation. Now, there are a lot of strategies that rely on biological treatment. And so, for instance, I did my PhD work on biofiltration, so thinking about how we could utilize microbes to degrade environmental contaminants that are volatile. And then also just wastewater treatment in general. There are methods that you can use, but the concept is really similar, where you're utilizing microbes to degrade XYZ contaminant. But under those scenarios, it's much easier, or I should say, not necessarily easier, but you have more control over the situation because you could dilute it, or you could find the proper conditions to optimize the degradation. But yes, biological treatment can be used for a wide range of treatment processes.
So are there a lot of regulations out there for companies that they need to start using this kind of?
So there are regulations in terms of certain contaminants that can't be released, and of course that'll be highly dependent on the situation. So let's say if you're manufacturing paint, or if you're so it's going to be it'll be very specific to that specific industry.
I have read that soil bacteria can be very difficult to get to culture in the lab. How do you deal with isolating and propagating something that you found in the soil so that you can use it someplace else in the future?
Yeah, so that's a great question. You are 100% correct. Organisms out in the environment are very, very difficult to culture in the lab. And the reason for that is that they often need the synergies with the other microbes that are present. And we have no way to replicate in our lab the actual conditions from the subsurface. So that's where a lot of these genetic techniques are actually great, because now rather than culturing them, we can actually get a snapshot in situ of what is actually happening. So we can now figure out, okay, here are the thousands of microbes that are present, and as we build our databases from all these different locations, we can start gaining an appreciation for which microbes actually like to live together. And as we start really screening through the genomes of all of these different microbes, we can start understanding what are the particular functions that we need to be stimulating at the same time. So that's really the basis of fundamental approach that that really this field is moving towards, because you're entirely right. We can't culture a lot of these, or you might culture the wrong thing. That's the other aspect to the question.
So my question, I guess, is about magic bullets. Locally in our drinking water in the Triangle, there was efforts to remediate nutrification of our drinking water system through sort of biomechanical systems, and they were a bust. And so I'm wondering what you feel about a purely somewhat purely not mechanical but biological solution to pollution. Is that, how do you feel about that being, other people thinking that that's the solution, that we can do it that fashion, that it's more closer to nature, et cetera, et cetera?
So, of course, being that I'm a biological engineer, I always think microbes have the solution to everything. And I think in many cases that you can rely on microbes to degrade contaminants. Now, there are a wide range of contaminants, and metals would be the perfect example, that you can't remediate because you might transform them into another type of metal that might have different properties. It might you might be able to stabilize it in one way. So I would say that for some situations, certainly a biological approach would be feasible. There are certain cases where it will not be the most optimal solution, and so I mean it's always important to look at both of them. So as much as I would love to say bacteria or microbes will always have the solution, unfortunately I can't go there, at least not while I'm on TV. So
so you mentioned earlier introducing like flash fire kind of like microbes that are poorly situated for the environment but feed only on the certain contaminant you're looking for. And do you think what are the pros and cons of using that technique versus taking existing microbes from the area and introducing the trait to also be able to consume your pollutants?
So if I'm understanding correctly, you're saying what are the pros and cons to introducing a foreign microbe as opposed to really stimulating? So the biostimulation versus bioaugmentation types of approach. Yeah. So biostimulation should always be the first approach. It'll be much easier if you have a native microbial organism that has the capacity to degrade your contaminant and is present at your site. And the reason why that is a much better approach is that organism has adapted and it can compete for resources under those specific conditions. When you're introducing a foreign organism, you really have to be careful in terms of what that's going to do to that native microbial population, and really thinking about, are you actually going to be able to help that organism really compete with the other ones. So one of the dangers of a bioaugmentation approach is one that it's just not going to grow, and so it'll just be wasted. The other one is it might be able to degrade one of the contaminants, but maybe you have a suite of contaminants, and so it might outcompete one of the other microbes that you actually need. And so you really have to be careful about introducing that foreign organism.
Thank you very much for the talk. You mentioned earlier that the outcome of the process of remediation could be carbon dioxide. I don't know if that was just an example, but I guess my question is just unintended consequences in general, which you kind of just answered. I mean, if they're already sort of local to that environment, probably going to be fewer consequences. But in the case of, the fact that maybe some are emitting carbon dioxide.
Yeah, so one of the unintended consequences is that you might have some incomplete degradation products that happen. So a much better scenario would be, right, that it's incorporated only into some type of carbon or nitrogen that can then be recycled and reused by another organism. There are scenarios where we actually make compounds that are more toxic than the parent compound. And so the perfect, I guess the poster child for that was really for many years, we thought perchloroethylene or trichloroethylene, which are degreasing agents that were also used in dry cleaning industry. And so for those of you that are chemists, this is a double carbon with an ethene bond and then 4 chlorine atoms around them. And the sequential removal of each of those chlorine chlorine. So we have perchloroethylene, so 4 chlorines that goes to 3 to 2 to 1. And the one where you have 1 is known as vinyl chloride. And so vinyl chloride is a carcinogen, and it stays, it sticks around for a really, really long period of time. And so for many years, this was kind of the poster child for why we didn't want to stimulate bioremediation. We later found out that actually you can degrade vinyl chloride and it turns into something that is non-toxic. But that's the worst nightmare, is that you make something that's more dangerous than what you started with.
So do you have like a favorite project? If somebody said to you, what's the best advertisement for the bio approach to this one that's been a huge success, what would you pick?
Did you say what is my favorite?
Yeah.
My favorite
Your biggest
project.
success, yeah.
So I'll say that the precision bioremediation approach that is the best studies is the perchloroethylene and the trichloroethylene remediation. So while I wouldn't say I have a specific location or a specific site, that is the one that we have the best understanding of how to actually engineer a bioremediation strategy. So I'll stick with that.
So I'm curious about this picture on the bottom of your slide, and what sort of like I can imagine working in the lab, that's one thing, but actually working with these microbes out at a Superfund site has its own set of hazards. So how much field work do you do, and what sort of precautions do you take?
So that's a great question. So I got that picture off the internet, so I don't actually know what these people are being exposed to. But that being said, so when we go out in the field and actually collect samples, I mean, we need to be very careful with what we're being exposed to. So my students, it's been a while since I've actually gone out and sampled, but my students would have this type of personal protective equipment, or some big boots, or some type of gloves or something. But safety always comes first.
So most of the examples you've given are examples of precision bioremediation and dealing with challenges that are localized toxic sites. But is there any hope of ever being able to use these kinds of methodologies to address big, broad, global climate type problems?
So I would like to believe that there is. So one of the ongoing projects that we have is actually looking at how microbes are being transported in ballast water. So I don't know how many know what ballast water is. Ballast water is the water that's used on ships to actually balance them. And so we're trying to understand, when there's ballast water that's traveling around the world, how if you bring water from a tropical environment and you release it in a very different environment, how that is affecting that microbial ecology. And also thinking about antibiotic resistance genes, and so trying to start gaining an appreciation for how genes are being transferred in general. So eventually, there's a project called the Earth Microbiome project that's ongoing, and many scientists are part of that effort. But the idea is really to be able to really map microbiomes in general, so that we can understand if there are specific inputs into that ecosystem or that environment. What does that trigger? What is the long-term impact? So it could be thinking about it, as you mentioned, right? Thinking about ecologically on a global scale, or maybe it's thinking about an algal bloom, or thinking about things that are bigger than just at a given site. But it's a big problem, and it's going to take many scientists and a lot of research before we get there. But hopefully, eventually we'll get there.
Speaking of global scheme, so we're in the midst of the Olympics right now. So who's in the gold position, who's in the silver, and who's in the bronze position to lead in this field?
Duke University.
Oh, Oh man, you want names? Or you want? So I think there's a lot of us that are looking at this challenge, I would say. So in the field of bioremediation, we're really just starting to gain an appreciation for how we can model these different interactions, and so I'd say there's a lot of people that are at the same level in environmental engineering right now, where we're really making advances in precision medicine or in applications that are related to the human body, because there's so much more data that are available in that system. And so my hope is that, we're going to get there too. It's amazing to me, and I mentioned this earlier, that you can tell, we know what 99% of the microbes in somebody's gut are, but you go out to a field, and even out here, if we were to collect soil sample, you might know what 40% of those microbes are. And so we still have a long way to go. We have to first figure out what are those other 60%, and then we have to figure out what can those 60% do. So it'll still be a while, but I don't know who's in the gold. Maybe Duke, like you said. I'd like to think of, that I'm a leader in that field. So,
Hi. I saw an article recently about some microorganism that actually excretes gold particles. Is that something viable that might be used for turning, say, hexavalent chromium into tetravalent chromium?
So there are, so I don't know specifically which study you're referring to, but there are biogenic formation of nanoparticles. And so I know this is possible for silver, for instance. So I could imagine that that's similar. So there are many different strategies that can be exploited. And so I don't know enough about chromium to be able to speculate, but certainly there are ways to engineer or to utilize microbes to carry out particular functions.
Yeah, hexavalent is carcinogenic and tetravalent is not.
Yeah, I was just saying I don't know with the gold specifically what the interaction, so, but yeah, microbes can they're always a good place to go, or start at least.
You said that you were interested in finding the indigenous populations first. I'm wondering whether you have ever seen a population mutate into be more useful over time.
Yes, so that's a great question. There are microbial communities themselves that will evolve over time, meaning that the composition, some microbes will be more and some will be less. And then individual microbes themselves over time are going to evolve. So so we've seen it in the lab. Those happen if you can grow them fast enough. Certainly, that is a possibility, and I'm sure it's happening in the lab or in the field as well.
Let's give Claudia one more round of applause. Thank you very much for being here tonight. Thank you very much, and of course, thanks to all of you for coming out and asking awesome questions. One of the most impressive things about the Science Cafe are the questions, the Q&A part, when we find out what you want to know about the topic of the night, and then just how smart you people really are. It's so exciting to have these kinds of awesome discussions about what's going on at the cutting edge, at the frontier of all sorts of science, technology, engineering, mathematics, even sometimes art and humanity disciplines. So thank you to all of you for coming out to the Science Cafe. Next week here at the Cafe, just to give you a heads up, we're going to be doing a little bit something different. Next week's Cafe is called In Between the Science. I've actually booked 4 different specialists who are going to come up and talk about their work as it happens in between their professional work. So what's it like being a scientist? So it's going to be sort of a storytelling night. So come out, get dinner, get a drink. We're going to hear some awesome stories from 4 incredible scientists, and I hope that we'll see you next week at the Science Cafe. Good night, everybody.